That is the end of Part B. Now look at Part C. Part C. In this part of the test, you'll hear 2 different extracts. In each extract, you'll hear health professionals talking about aspects of their work. For questions 31 to 42, choose the answer A, B, or C, which fits best according to what you hear. Complete your answers as you listen. Now look at Extract 1.
Could you please introduce yourselves and give us a brief overview of what work involves at Thermo Fisher Scientific?
We both work within the field and safety instruments. Our latest instrument is the Thermo Scientific Ramayana Process Analyzer, which makes process monitoring and optimization accessible, practical, and profitable. For some time now, we have been producing and supporting handheld instrumentation to perform incoming raw material ID and realize the maximum efficiency of a customer's incoming material ID and verification scheme. Our goal has always been to keep technology and instrumentation easy to set up and use, and retain robustness and field survivability to make our solutions more effective than current or historic testing methodologies. Our products empower and enable you to make rapid, trusted analytical and business decisions. We believe the Thermo Scientific Ramayana Process Analyzer enables real-world cost savings you can expect when using Process Raman, as Thermo Fisher Scientific can help users achieve their process analytical technology strategic goals within the biopharma space.
What is process analytical technology, and how is it best implemented?
Process analytical technology covers a compelling area for us as an organization. It allows us to deliver clutch workflow and streamline process solutions within the biopharma space. Using process analytical technologies opens up a world of insight into a given bioprocess and provides the means to tackle problems as early as possible. Process analytical technology is not a new concept. Most larger companies are leveraging process analytical technology solutions at some point or another in their pilot and manufacturing plants. We believe that there is no reason why smaller, mid-sized biopharma companies cannot also leverage similar solutions. Process analytical technology can be used in several manners, such as offline or outline analysis, which requires sampling, and some automated analysis instead of manual wet chemistry. PATs may also be used as online and inline systems where the need to sample is eliminated. Process analytical technology is important for this industry. It matters greatly when challenged with tightening a process for FDA approval, when there is a strong need to beat out the competition in the market with rapidly developed therapies, or even when your goal is to implement practical process improvements and ensure product yields and costs are kept under tight control. We are well aware of some of the challenges faced by those in the biopharma field when implementing process analytical technology solutions. The end user needs to harness data for improved process understanding and control in real time. However, This is difficult when you need to take time to sample for offline analysis and risk potential contamination. It is even more frustrating when there are struggles with getting a newly acquired process analytical technology solution implemented in your flow. Users also need to be quickly and properly trained.
What is the value of having process analytical technologies available?
There is a clear value in having a process analytical technology solution, particularly one that can give you real-time actionable results while minimizing or eliminating contamination hazards. The real question is, why should you care that your data is available in real time? Just ask yourself, how much time is wasted when something goes wrong? It is extremely important to get things right the first time. Anytime you have to carry out work again is considered waste. How much money do you stand to lose because you did not have the right data to make a quick decision? It can be extremely difficult to recover when trust is compromised, especially with how rapidly information can travel and be shared in the modern era.
How can Thermo Fisher Scientific's technology help monitor inline R&D biopharma processes?
Our process analytical technology leverages Raman spectroscopy to see real-time changes. We speak with our customers directly and understand the urgent need to monitor inline R&D biopharma processes, both upstream and downstream. When a customer is equipped with a process analytical technology solution that enables a rapid knowledge infusion about any given process, it is then possible to closely control that process and boost the capacity of the existing development team in ways other than just headcount increases. Our customers can make those critical pivots when they have real-time access to actionable data. Our team at Thermo Fisher Scientific is equipped to provide expert assistance with implementing these process analytical technology solutions.
What current process analytical technology solutions is Thermo Fisher Scientific offering?
We have developed the Thermo Scientific Ramayana Process Analyzer, a small, extremely portable, robust Raman spectrometer. Raman spectroscopy uses laser light to generate spectral data that may be interpreted to determine target and light concentrations and identities. It is non-destructive and very specific when performing qualitative analysis. The presence of water has a negligible impact, which is ideal for aqueous matrices. Using an immersion probe and a fiber optic cable, Ramayana Process Analyzer can provide in situ bioprocesses measurements. A range of accessories is also available, such as a flow cell, if non-invasive analysis is preferred. Our extensive Raman portfolio includes Ramayana Process Analyzer, the XR3 Raman Flex system, and our handheld TrueScan RM handheld Raman Analyzer.
How and why is the new Thermo Scientific Ramayana Process Analyzer useful?
The Ramayana Process Analyzer is both small and not over-engineered, while the system is very intuitive as the user interface reduces the complexity of analysis and setup. Within 15 minutes, Ramayana Process Analyzer generates real-time actionable data for critical process parameters right out of the box. Ramayana Process Analyzer also enables better decision-making that impacts the bottom line by transmitting information within seconds or minutes rather than hours or even days, which is seen when using legacy techniques. The Ramayana Process Analyzer system is useful for obtaining rapid answers to key questions relative to bioprocessing applications where time to results is critical. For instance, potential application uses include metabolite analyte monitoring, such as looking at glucose lactate levels for upstream applications, and protein product purification in downstream processes. A couple of other applications include target product concentrations and cell density measurements. These are all applications where you must get the correct result quickly. Ramayana Process Analyzer allows users to collect and analyze spectra within seconds and tightens up process control parameters quickly.
Does implementing a process analytical technology solution, such as Ramayana Process Analyzer, positively impact manufacturing processes?
Yes, it does. For example, we recently had one real-world use case of the Ramayana Process Analyzer system compared to a legacy automated offline analyzer. This study was performed over 4 working weeks and examined the cost of upstream batch monitoring of a mammalian cell line. Using Ramayana, critical process parameters, such as glucose, pH, gas analysis, VCD, and cell viability, were all monitored using a single system. This approach facilitates rapid real-time eye-in-the-sky monitoring of bioprocesses with real positive outcomes. Cost-wise, we are talking about $80,000 in savings annually for glucose alone. That compounds into a $300,000 annual savings when using other critical process parameter tests that are eliminated because you are using RAMINA process analyzer. Armed with this knowledge, decisions on how to best optimize R&D headcount and instrument resources, including the resultant waste of those resources, can be made easily, especially when you are performing process improvements and know your current cost of business. Leveraging the unique nature of RAMINA Process Analyzer as a small, easy-to-use, and accessible Raman process analytical technology system creates a compelling business. Thermo Fisher Scientific can also be leveraged as a trusted source and resource for assistance.
Could you provide an overview of the type of performance and application utility users can expect from RAMINA Process Analyzer?
RAMINA Process Analyzer can be used for various applications in the biopharmaceutical industries, including for upstream and downstream processes, such as the purification of proteins and nucleic acids, or even for identification and characterization of finished products. There are challenges when monitoring and running conventional processes in a bioreactor. Bioreactors are used extensively throughout the biopharmaceutical industry, for example, during the manufacturing of monoclonal antibodies, and RAMINA Process Analyzer can help overcome some of these challenges.
What are the main challenges when monitoring and running a bioreactor?
The process of monitoring a bioreactor requires someone to specifically pull the samples and then run it to offline or outline measurements. This involves resources and time. There are critical challenges with this process. For instance, there may be a fluctuation of metabolites in this process. The metabolites can then vary dramatically in a bioreactor, or a bioreactor might sometimes go in the suboptimum of operating conditions, which may ultimately lead to a lower quality product. Another challenge encountered when running a conventional bioreactor is contamination. When pulling a sample, the aseptic bioreactor is in contact with environmental factors and increases the risk of contamination. At Thermo Fisher Scientific, we tend to come up with solutions for these problems based on Raman technology, which is where the RAMINA Process Analyzer comes in.
Now look at Extract 2. Extract 2, questions 37 to 42. You now have 90 seconds to read questions 37 to 42. The COVID-19 pandemic has created unprecedented challenges for healthcare services, including the delivery of FCC. As part of a larger, multinational, prospective, observational cohort study on the epidemiology of COVID-19 in neonates and children managed by NICU and PICU services, we collected data on FCC practices for neonates born to mothers with COVID-19. At the start of the pandemic, rooming in and maternal breast milk feeding rates were low, but both increased over time. To our knowledge, this is the first study to assess temporal changes to FCC at a multinational scale. Our findings suggest that as the understanding of the risks of COVID-19 to neonates evolved, so did practice. Physical contact was highly restricted for neonates prior to separation from their mothers, irrespective of the reason for NICU or SCN admission. This highlights the ongoing challenges the pandemic poses for the delivery of FCC. We found that of those neonates tested for SARS-CoV-2, 5% had a positive result. This incidence aligns with a living meta-analysis indicating that the SARS-CoV-2 positivity rate in neonates born to mothers with COVID-19 is as low as 1.8%. Our incidence was higher than a UK population-based cohort of perinatally exposed neonates. 17 neonates acquired SARS-CoV-2 infection, and the 0.9% of infants born to SARS-CoV-2 positive mothers in a nationwide cohort study in Sweden. This likely reflects our NICU-focused design, which was limited to high-risk perinatal hospitals within a research network and the lack of testing in 116 neonates. Early in the pandemic, case reports of symptomatic cases in neonates born to mothers with COVID-19 emerged. Fortunately, as the pandemic progressed, it became clear that the severity of neonatal disease is usually mild. This is a finding we also identified, with 52% of our SARS-CoV-2 positive neonates being asymptomatic, consistent with a meta-analysis of neonatal SARS-CoV-2 infections reporting clinical features of COVID-19 in half of patients. Many symptomatic positive neonates were also born preterm, making it difficult to differentiate the cause of respiratory failure and other symptoms. Although perinatal transmission is rare and generally asymptomatic when it occurs, our study has shown that the provision of FCC may be impacted for any neonate born to a SARS-CoV-2 positive mother, independent of neonatal infection. We therefore promote the implementation of FCC practices into infection control measures during the future stages of this pandemic to provide neonates and mothers with the best health benefits. Our study identified that only 46% of mother-baby diets roomed in together. Local reports of rooming-in practices at the start of the pandemic were varied, ranging from 13% to 83%. In our cohort, rooming-in was unlikely to have influence rates of neonatal infection. In contrast, a meta-analysis found that rooming-in was associated with a risk of delayed neonatal SARS-CoV-2 infection. Neonates can be separated from their mothers for other reasons, principally the need for NICU care. This was present in our cohort, with more separated neonates being born preterm or to mothers with perinatal complications. The unnecessary separation of the mother-baby dyad is associated with negative outcomes for both neonates and mothers. Thus, it is concerning that in our cohort of neonates, in whom only a small number were infected with SARS-CoV-2, 73% of those separated from their mother were admitted into a NICU or SCN without any symptoms or comorbidities to account for admission. This may impact NICU access for other neonatal conditions. It has been suggested that high NICU admission rates may be secondary to the logistical challenges of isolating neonates in a postnatal ward setting. Our data, however, showed that it was rare for an asymptomatic neonate to be managed in a NICU due to lack of postnatal ward capacity. The onset of the pandemic varied across regions, but FCC guidelines appeared consistent within participating sites, with most recommending rooming in for all the study period. Whilst our data does not allow evaluation for the cause of separation, the increase in rate of rooming in within sites that had an active policy to do so likely indicates that COVID-19 status did influence separation practices early in the pandemic. With time, clinicians became more familiar with managing COVID-19 in a perinatal population. FCC rates were highest when there were the lowest rates of moderate to severe symptomatic COVID-19 in mothers and symptomatic disease of any cause in neonates. It is therefore also possible that FCC increased due to maternal and neonatal clinical manifestations, not due to changes in practice. We observed a decrease in rates of rooming in and maternal breast milk feeding from April-June 2021 after a clear trend of improved FCC practices. This epoch coincided with the emergence of the Delta wave over the Northern Hemisphere, which is known to cause more severe disease in mothers. Although a smaller sample than the preceding epochs, our data support this and suggest potential additional neonatal implications. Rates of moderate to severe symptomatic COVID-19 in mothers, cesarean sections, prematurity, and neonatal ventilation increased in the epochs following April-June 2021. There were no changes to guidelines, which all recommended vaginal birth, where possible, for pregnant women with COVID-19 in 2021. Unfortunately, we did not document the strain of SARS-CoV-2 or have access to mother-baby diets without perinatal SARS-CoV-2 infection. The latter limits our ability to determine whether the changes in FCC were specific to maternal infection status or do restrictions applied to all mothers. We also do not know how the impact of new viral strains after October 2021, such as Omicron, in a population that is mostly no longer immunologically virgin impacts perinatal transmission and eventually FCC practices. Another striking finding was the lack of mother-baby physical contact prior to separation in almost all neonates who were separated. This translated to almost half of all neonates being denied early and close contact with their mother, even though most sites had a general approach to allow it. This may be confounded by the fact that separated mothers and neonates were more unwell than those who roomed in, so may have been unfit for SSC. This may also, however, represent infection control measures overriding FCC recommendations. In an online survey of mother-baby physical contact in the NICU during the COVID-19 pandemic, only 20% of parents had some contact with their newborn. As the risk of perinatal transmission is low and neonatal COVID-19 is generally mild, it is imperative that health services prioritize early and close SSC with appropriate hygiene precautions, as per current recommendations. A limitation of this study was that we did not collect data on physical contact after initial separation and the use of hygiene precautions during contact, like masks and hand sanitizer. The benefits of infant feeding with maternal breast milk are well understood. It was concerning that only 52% of neonates in our cohort received breast milk from their mother. This rate is lower than previously reported in localized studies during the pandemic and the generally accepted rates in large NICU populations. Rates of maternal breast milk feeding in our international cohort are unlikely to be uniform before the pandemic and may have been influenced by maternal, neonatal, and cultural factors, as well as limitations on NICU visiting after maternal discharge from birthing hospital. We contend, however, that it is likely due to study design. Epicenter was designed to capture populations managed by neonatal clinicians, which will be skewed towards sicker neonatal and obstetric populations, in which maternal breast milk exposure will be less. Previous reports of feeding practices were derived from obstetric cohorts or population-wide data. Despite this, It was reassuring that maternal breast milk feeding increased over the pandemic from 23% to 70% by January-March 2021. This temporal change in maternal breast milk exposure rates is likely due to health services adapting their policies and increased clinician comfort in response to data showing no association between breast milk and positive neonatal infections. Although our study was not designed to address infectivity, we also found no difference in neonatal infections between the neonates who received maternal breast milk and those who did not. Only 22.4% of neonates born to mothers with moderate severe COVID-19 disease at birth receive any breast milk, despite breast milk being broadly promoted. Promoting breast milk feeding is not the same as achieving it when there are maternal comorbidities. Our study demonstrates that it is possible to maintain lactation, even in mothers with severe COVID-19. This provides invaluable insights into breast milk exposure more broadly for neonates born to moderate severely unwell mothers of any cause. Reports of lactation support, and conversely suppression, during postnatal maternal illness are sparse. We would suggest that neonatal, obstetric, midwifery, and if appropriate, infection control clinicians collaborate to advocate for stimulation of lactation when caring for neonates, irrespective of severity of maternal illness. There are several other limitations to this study. This is an observational study limited to participating NICUs across different regions with different healthcare models and cultural influences. This may cause differences to the approaches to FCC. The similarities in available guidelines during the pandemic and the increases in FCC as the pandemic progressed suggest that maternal COVID-19 status had a major impact on FCC. Whilst all hospitals embrace the same core principles of FCC before the pandemic, we do not have data on pre-pandemic implementation rates. This is a limitation broadly applicable to the field of FCC, as high-quality data on implementation of FCC is rare. Our study demonstrates the importance of collecting these data and feasibility of doing so. That is the end of Part C. You now have 2 minutes to check your answers.